Hi class, this is your instructor, Skylar Huff. I'm back now, and we're still here in the 10th chapter. One thing I want to ensure that you know is that just because these videos have been made, it does not mean that there is any substitute for reading your textbook and taking notes. So please, as you listen to this, have your very own textbook out, have your paper out for taking notes, and I would say also have the PowerPoint presentation somewhere nearby. That way you can refer to it. You all should ensure that you all review. In saying that you all should review, I'm referring to reviewing neurons. I say neurons because a neuron can be classified in more than just one way. We did this a little bit before the break, meaning spring break, but I want to make sure I reiterate that you should know those structural classes of neurons. When I mention that, I'm referring to that unipolar neuron. And when I say unipolar neuron, I'm meaning that that neuron has one process that extends from that cell body, and of course it forms the central and of course peripheral processes, and those together comprise the axon. Along the same note, you all should know the bipolar neuron. It has two processes extending from the cell body. One is a fused dendrite, and the other is an axon. It's in the name. And then lastly is the multipolar neuron. And that multipolar neuron is a neuron that I typically draw on the board. It has many processes that extend from the cell body. All are dendrites except one single axon. So to contrast that, I'll say it this way. What you should think about is that the multipolar neuron is that most abundant type of neuron in the body. And it's a major type in the central nervous system. The bipolar neuron, it's pretty rare. It's found in some special senses, such as the ear, the eye, and the olfactory mucosa. And then lastly, the unipolar neuron is mainly found in the peripheral nervous system, being part of that dorsal root ganglia of the spinal cord and the central ganglia of cranial nerves. Secondly, what you all should know is the functional classification of neurons. When I speak to that functional classification of neurons, I'm speaking to the neurons that are referred to as being sensory neurons or afferent neurons. Those, of course, are conducting impulses from sensory receptors in the skin or your internal organs toward or into the central nervous system. So as I stated that way, you all should know this like the back of your hands. We'll get to the special senses as soon as we finish this chapter. That'll be chapter 12. The next type of neuron is the motor neuron or efferent neuron. And in this case, it carries impulses away from the central nervous system to effectors. And I refer to effectors as being either a muscle or a gland. Lastly is the interneuron or association neuron. And these are what lie between a motor neuron and a sensory neuron. And of course, they're taking that impulse through that central nervous system. So as I mentioned this, most of these interneurons are located within the central nervous system. So please keep this in mind, especially as you get to your test. Because in your test, I'll have a picture of the reflex arc, and I expect you all to know each and every part. As I get to what comes next, make sure you all go through those clinical applications in the textbook. I won't spend a lot of time on those throughout this lecture, but here I am at multiple sclerosis. As I get to MS, called multiple sclerosis, what happens is these scleroses form. And when I mention those scleroses are what cause, I guess I'll say, a break, or even stop, or even slow transmission. Meaning, without proper input, what can occur is, of course, what's involved in multiple sclerosis. So, as I get to it, it states that what happens is it's likely going to be something of the effect of the person's own body. Meaning, this is when the body attacks itself. It's an autoimmune condition. So, as the myelin sheath is attacked by that person's antibodies, it's destroyed. And what you all have learned by now, I hope, does help, In other words, when this occurs, the myelin sheath is destroyed, the sclerosis or scar is left behind, which will stop, which will impair transmission of nerve impulses. So as that occurs, it would definitely, of course, stop contraction of muscles and, of course, causing atrophy to ensue. And I'm referring to muscle atrophy here. Otherwise, a person may exhibit a bit of numbness or even weakness in limbs, blurred vision, and even fatigue. Please make sure you all know about multiple sclerosis for your test. And of course, to be treated, immunosuppressant drugs would work well because, of course, this being an autoimmune condition, by suppressing immune activity, of course, it should help. So with multiple sclerosis, it's quite painful, meaning I would even say debilitating pain is experienced in a person with multiple sclerosis. So as the myelin sheath is destroyed in the central nervous system, they become non-functional. So as it happens, the axons themselves are not damaged. And even though it's not damaged, it says there will be a growing number of sodium ion channels to appear in that demyelinated fiber. So of course, what then occurs typically is going to be blindness that may ensue and impaired speech. So as mentioned earlier, it would help to, of course, reduce the complications, and it in hopes, of course, of getting to some, I guess I'll say, cure, to, of course, get that myelin sheath to form again. So this shows, of course, the classification of neurons, this, of course, being structurally, with the multipolar from left to right at A, bipolar at B, and C, the unipolar neuron. Next up, of course, it refers to those sensory neurons, which I just mentioned. Those sensory neurons, which I did just mention. And as I do mention that, just keep in mind that this is intricately important to where we're going from this point forward in the nervous system. So upon finishing the classification of neurons, we'll now move on. So at some point in life, there very well may be damage. So what I'm getting at is I mentioned a long time ago that we have cells that can divide, and then there are cells that cannot divide. If you take your cells back to, of course, what you all learned about tissues, as I got to nerve tissue in teaching, I said that, remember, that nerve tissue is, of course, overall considered to be amitotic. I say amitotic because, of course, the typical neuron does not divide. So if in fact the cell body is damaged, that would, of course, mean that the neuron would typically no longer function and die. So as I get to the way in which neurons are damaged and get to regeneration, what I'm getting to is in the central nervous system. Yes, I know I skipped the peripheral nervous system. In the central nervous system, regeneration is quite unlikely, meaning it's happening because the oligodendrocytes, they do not proliferate. They do not, of course, I'll say, persist after the injury. And, of course, don't look for regeneration. Hence, of course, that's why they say in the event that someone has had a serious injury, I refer to an injury class of the spinal cord or injury of the brain, that's when things become quite dire. On the converse, I now speak to regeneration in the peripheral nervous system. So in the PNS, if that peripheral axon is injured, it may very well regenerate. Of course, it says that an axon separated from the cell body and its myelin sheath will degenerate, but as long as it's still attached, it should regenerate. So, of course, the Schwann cells and the neurolemma remain. So those remaining Schwann cells provide that sheath, meaning that myelin sheath, for that growing axon. So, of course, if the axon establishes that connection that was once there, function comes back. However, if that axon cannot connect to, of course, what was once there, function, of course, may very well be lost. Keep that in mind, of course, because that too will be found on your test. So as it states here, you have the site of the injury, in whatever case it may have been. So with that injury, you have Schwann cells, I'll say downstream. I say nearer here to that effector, the skeletal muscle. So it states that the distal portion of the axon would degenerate. However, that proximal end, nearer to the site of injury, shall regenerate. So what then happens, of course, is the Schwann cell tube extends distally, and thereafter the Schwann cells form that new myelin sheath, and of course what was once connected is reconnected. This is in the peripheral nervous system, not the central nervous system. So what I'll now get to are factors that affect impulse conduction. So I state this because these ions are integrally important to the conduction. So it states if there is an increase in the concentration of potassium ions outside of the cell, it will reduce that potassium gradient. By doing that, threshold will be reached, of course, with stimulus of a lower intensity. To say it another way, the neuron becomes excitable, and it's more excitable and takes less of a stimulus to excite that same neuron. If there is a decrease in potassium in extracellular fluid, the neuron can become hyperpolarized. And with this, I would hope for those who know, you already know what I'm going to say. That means that an action potential will not be generated, and will lead to, of course, that lack of impulses and muscle paralysis. And even so much so that it would take, of course, a stronger stimulus than that original stimulus to elicit an action potential. And then lastly, a decrease in potassium ion permeability can be caused by some anesthetics. I mention this because anesthetics are those that can stop the impulses from passing through that tissue around the axon. So with this happening, the impulse will not reach the brain, and of course you cannot perceive pain. It's quite amazing how pain can relate right back to these very same ions taught about in the chapter on basic chemistry. Now on to synaptic transmission. As I get to this, I alluded to this before we left for the break as well. Spring break I'm referring to. So here, sometimes, of course, as that synaptic transmission occurs, the nerve impulse will go from neuron to neuron to neuron to neuron, and thereafter, before it gets to that post-synaptic neuron, that message that was once electric becomes chemical before it can become electric again. So release of a neurotransmitter across that synaptic cleft, and of course it then binds to receptors on the membrane of that post-synaptic neuron. As I mention this, please, I repeat, please keep in mind the effect of neurotransmitters is not always going to be the same. I paused for emphasis. Sometimes that effect will be, of course, excitatory. In other times, class, that effect will be inhibitory. And it's based upon whether or not the ion channel opens or other ion channels close. This is a big key to understanding. So it states, of course, the chemically ion channels respond to neurotransmitters as well. So with that, from the changes in those chemically ion channels, we get these synaptic potentials. As I mentioned these synaptic potentials, what I'm referring to here is the way in which, if in fact there is an increase, I'll repeat, if there is an increase in permeability of sodium, I think for those who have studied and understand where we're going, you know what I'm going to say. If it increases sodium permeability, that of course will cause that membrane to become, I guess I'll say, less and less negative, or more and more positive, getting nearer and nearer to threshold potential that approximate negative 55 millivolts, increasing the likelihood of an impulse being generated. As on the converse, in the event that an inhibitory neurotransmitter moves the membrane farther from threshold, meaning becoming more and more negative or less and less positive, that will definitely decrease the chances of eliciting a nerve impulse. These two keys are important to know, especially for where we're going next. So with the excitatory postsynaptic potential. Excitatory postsynaptic potentials. So that just means that the membrane in which the neurotransmitter opens the sodium channels. I repeat, the membrane change in which the neurotransmitter opens sodium channels. So when those sodium channels open, of course, sodium rushes in, depolarizing the membrane as the sodium enters. And of course, the action potential becomes so much more likely. On the converse, it would be the inhibitory postsynaptic potential. This membrane change, of course, result of the neurotransmitter, will open potassium ions. So as those potassium ions open, and it could also be chloride ions, it hyperpolarizes the cell, getting more and more negative as the potassium leaves the axon, and of course decreasing the chances of an action potential. So next up, of course, is summation. It just states that adding both of those together, the excitatory postsynaptic potential and the inhibitory postsynaptic potential, gets us to what is called summation, meaning that effect of course, both of those. So now we'll get to neurotransmitters and their actions. As stated by your text, it's an estimated 100 neurotransmitters. I'll help you out with this. Acetylcholine was the first to be discovered, and of course, not just the first to be discovered, but the most well-known neurotransmitter. So it's been studied, of course, and it's a lot easier to study because it's released at neuromuscular junctions. So neurotransmitters can be classified in a number of ways, such as being monoamines, amino acids, and even peptides. So of course, as they are produced, they're produced, of course, in the rough endoplasmic reticulum or cytoplasm. And when the impulse reaches that synaptic knob, that synaptic terminal, the end of the axon, the neurotransmitters are released by way of exotosis. Now, what causes that release? Please think about it. What causes that release? Their release, of course, is fueled by the influx of calcium ions, and was all predicated on the change in voltage. So those voltage-gated calcium ion channels open up, calcium floods, increases in concentration, vesicles of some neurotransmitter, be it acetylcholine, norepinephrine, dopamine, serotonin, or even histamine, and of course it was released by way of exocytosis across that synaptic cleft. So as we get here, in your test, I'll ask about those neurotransmitters as they're classified by type. So I'll begin here quickly with neurotransmitters. In the CNS, it controls skeletal muscle actions, and of course, in the PNS, it says it stimulates skeletal muscle contraction at neuromuscular junctions. So please remember, we won't have a test for, I would say, about 2 weeks, but of course, it says at neuromuscular junctions. You will explain the events occurring at the neuromuscular junctions. I'll change my step here. However, it may excite or inhibit at autonomic nervous system synapses. So this is the key. It is all receptor dependent. This is why it's so important to know the receptor. So let's continue to classify neurotransmitters by structure. So I'll next get to those called biogenic amines. The biogenic amines include, of course, the catecholamines, such as dopamine and norepinephrine, and even epinephrine, as well as the indolamines. I did say that. So we have the catecholamines as well as the indolamines. The indolamines includes serotonin and histamine. So I'll begin here with, I guess I'll go down to norepinephrine. Its effect can be excitatory or inhibitory. It just depends on the type of receptor. I'll put it this way, though. In the central nervous system, it is referred to as being, of course, the feel-good neurotransmitter. I can't say it any other way. If it feels good, it is due to norepinephrine. Whether it's food, driving a car, coloring, going for a run, or even learning anatomy and physiology, it is all based on the release of norepinephrine by way of the brain. So I'll tack on what else occurs. What else occurs is in the event that someone uses an amphetamine, the release of norepinephrine is increased. And of course, removal of norepinephrine from the synapse is blocked by the tricyclic antidepressants, as well as cocaine. I repeat, if in fact someone uses cocaine or some tricyclic antidepressant, it ensures that norepinephrine remains on its receptors. So I'll put it this way, it's so amazing the way in which these chemicals affect. So I'll take it to a bit of a case here. In this case, it's about sex, drugs, and rock and roll. Hey, eat, drink, and be merry, it states. But as I just said, why on earth do we do the things we do? Why do we find those things we do so compelling, class? Well, it's because of our brain, and of course the pleasure center of the brain. So the reward center that I'm referring to, of course, is all about what is called norepinephrine. So I'm going to introduce the second of which, called dopamine. Dopamine and its effects could be either A, excitatory, or B, inhibitory. It just depends on the receptor. So dopamine in the CNS will be, of course, released by way of the substantia nigra of the midbrain, not to mention the hypothalamus. It, too, is known as the feel-good neurotransmitter. I say again, it, too, is a feel-good neurotransmitter. So if you need help remembering these, think of the catecholamines, including dopamine and norepinephrine, the feel-good neurotransmitters. So as I stated that way, the release is enhanced by L-dopa and amphetamines. There it is again. And when cocaine is taken, reuptake of dopamine is, of course, blocked. Hence, that person or persons enjoy the drug and need more of that drug called cocaine. So if a person has Parkinson's disease, that means that they will be deficient in dopamine. And the last part I'll put here is that dopamine neurotransmission increases in schizophrenia. And if you say, wait, what do I mean? What I mean is emotional behavior is controlled by our biogenic amines. So that means that in the event that our motor neurons of the autonomic nervous system, as they release these catecholamines, if there is an imbalance of these, they're typically associated with a mental illness, such as overactive dopamine signaling occurs in schizophrenia, and even certain psychoactive drugs such as LSD. I did say LSD, or even mescaline. It can bind to those biogenic amine receptors and provide hallucinations. So to get back to, of course, that feel good, I'll put it this way. Ecstasy. The ecstasy of romantic love may just be a brain bath of neurotransmitters such as glutamate and norepinephrine. I haven't quite gotten to glutamate yet, but I'll get to it. It's one of those amino acids by class. So, of course, as it states here, the very same thing is that drugs can abuse. They can subvert the powerful system because these neurotransmitters of the reward system are chemical cousins of the amphetamines. So crystal meth, the methamphetamine, is an artificial stimulant. So it provides their brains with a highly addictive pleasure flush. However, it's so short-lived. And because the brain is flooded with those neurotransmitter-like chemicals, it makes less of its own. Why make it when I can take it? The brain thinks, I suppose. And the same was with cocaine, another reward system titillator. So it's been around since the ancient times. And it states that it was once a toy of the rich in its granular form and inhaled. However, a cheaper, more potent form, smokable form, called crack, is available to the masses. So for $50, somebody can experience a rush of intense pleasure. But of course, crack it's as treacherous and intensely addictive. So it produces not only a higher high than the inhaled form, but also a deeper crash that leaves that person wanting more and more and more and more. So it takes that reward system and squeezes it dry, so to speak. So cocaine produces that rush by hooking up to the dopamine reuptake transporter protein, and it blocks the reabsorption, so it stays there. So it remains in the synapse, dopamine does, and stimulates the postsynaptic receptor cells again and again and again and again. So this is why That sensation is accompanied by A, increased heart rate, increased blood pressure, and even an increase in sexual appetite. So, as I mentioned moments ago, the body then releases less and less dopamine, and the reward system goes dry. So, of course, the cocaine user becomes A, anxious, B, in a sense of being unable to experience that pleasure without that drug. So those postsynaptic cells become hypersensitive and sprouts what are called, hey, new receptors. So in a desperate effort to pick up dopamine signals. So this, of course, is what I call addiction. Look for this on your test. This is what I call that vicious cycle of addiction as it begins. So the addict craves cocaine, needing that cocaine to experience the pleasure, but using it suppresses dopamine release even more and more and more. So the out of control, desperate cravings are, of course, quite difficult to manage, and they begin jonesing. So traditional anti-addiction drugs take so long to reduce the cravings that users commonly leave treatment programs. So please don't try it. So one way to prevent the cycle of addiction is to just prevent cocaine from ever reaching the brain. One time can be enough, you all. So please don't try it. There have been great results with this. So in clinical trials, a vaccine can dampen those effects and reduce the addict's use of the drug. Another approach, of course, is by keeping out those highs and lows by leveling out brain dopamine levels. But of course, the craving has made some addicts very creative, and they call them home pharmacologists, willing to experiment with anything, with a cheap mixture of cold medications, match heads, and even iodine and acetone yields crystal meth. So again, quite popular. It wrecks lives and often explodes in home laboratories. You likely have heard of those. So many people believe that certain drugs are safe and innocuous, such as, of course, ecstasy, MDMA, they call it. Somebody may have mentioned Molly one time. So this targets the serotonin-releasing neurons. So the rush of pleasure and energy that the user feels is due to the release of serotonin. So as I mentioned, serotonin, what happens in its case, it's mainly an inhibitory neurotransmitter in that it plays a role in sleep, appetite, migraine headaches, and regulating mood. So if you have a drug that blocks serotonin uptake, it of course relieves anxiety and depression. So what I'm getting at is, as this occurs, it may damage those neurons and cause loss of verbal and spatial memory. And then, of course, depression, sleeplessness, and memory problems may be permanent consequences just for this pleasure. And I'll end this thing pretty briefly here with sometimes people who want a pure, effective, and safe drug. I say safe. And they say drugs of abuse don't get them on the street. They get them from doctors. I think you may know where I'm going, class. Even people who would never dream of taking an illicit drug can be caught in the addictive cycle of prescription drugs. Prescribed legitimately to relieve severe pain, they call it OxyContin or oxycodone. It was so widely abused it had to be reformulated to prevent abusers from crushing tablets and storing the powder or dissolving it in water and injecting it in solution. So, what happened is a couple of years later, it's found out that 24% of users had found ways to circumvent the new formulation and had turned to other drugs. So as a result, the use of heroin among OxyContin users had nearly doubled. So what I'm getting at is this. The changes drove users from a drug of known strength and purity to an even more dangerous street drug of dubious, unknown purity and unknown strength. So as I say this, I just hope that this has let you all know what the brain does in response to these neurotransmitters. I'll be here briefly, class, with the amino acids. So with the amino acids, I mentioned GABA. And as I say GABA, it's, of course, gamma aminobutyric acid, you all. It's a general inhibitory neurotransmitter. So it's here. And as it does, it's here typically for as an anti-anxiety drug, and even in barbiturates. So with this is a pretty important amino acid. And not just that, but also glutamate. Glutamate is generally an excitatory neurotransmitter. Notice it says amino acid, so it's important in learning and memory. But the same way, it says it's that stroke neurotransmitter. Glutamate is. So, of course, it says excessive release produces excitotoxicity and can, of course, cause the neuron to be stimulated to death. So, most commonly caused by ischemia. Yes, I say again, that's that stroke neurotransmitter. And then up next is what I call glycine. It's generally inhibitory. And I'll leave it at that. Next up are the peptides. So when I get to the peptides, I'll say it this way. The peptides, of course, it says are simply just strings of amino acids. But with this, I'll say we have substance P. It's an important mediator of pain. So it says, by contrast, endorphins, including beta endorphin and dynorphin and the enkephalins, these are our class. Pay attention. Our natural opiates. I say again, these are our natural opiates. The neuropeptides are. And you say natural opiates? Yes. They reduce our perception of pain under stressful conditions. There was once on the news here locally, a story of a gentleman who had his house broken into in Mobile County. He shot those individuals. They didn't stop. They left, got in the car, and drove away. But they were found not far down the road. I think it was at a Dollar General. Thanks a lot, natural opiates. So enkephalin activity increases dramatically in pregnant women in labor. I suppose that's why some may say they won't know, I guess you say, anesthetics. So endorphin release is enhanced when an athlete gets that so-called second wind. And they may even call it that runner's high. Additionally, it's stated that these pain-killing neurotransmitters remained undiscovered until investigators began to ask why morphine and other opiates reduced anxiety and pain. So what was found is they just attach to the very same receptors that bind to our natural opiates, and of course producing similar but stronger effects. Thanks a lot, endorphins and substance P. So having stated such, I don't know why a person would want to take a drug, any opiate, when your body synthesizes its own opiates. And what I'll get nextly to will be, since I'm at the peptides, I guess I'll go on down to somatostatin. For those taking anatomy too, pay attention. You should remember somatostatin because it's quite interesting to know about, meaning it's a generalized inhibitory neurotransmitter. And it's often released with GABA. So it's a gut-brain peptide. And of course, it inhibits growth hormone release. That is somatostatin for you. And what I'll get to nextly, another gut-brain peptide, would be cholecytokinin. I mentioned cholecytokinin. It's called CCK. It's an excitatory neurotransmitter. But it's involved, of course, in inhibiting appetite. Hence, of course, a person shall eat, does eat, and when he or she does eat, that is when they say, oh, I don't want any more food. Thanks a lot, CCK. The purines are next. So with the purines, the one I say you all know lots about would be adenosine triphosphate called ATP. I'll leave it at that, but it does release, of course, that sensation of pain. And even adenosine. So caffeine stimulates the blocking brain adenosine receptors. And of course, it is involved in our sleep-wake cycles. And of course, it dilates arterioles and increasing blood flow to the heart. And ending this would be the gases and lipids. So as I mentioned, the gases and lipids, I like to get to these because it's important to know. Hence, of course, nitric oxide, the gas, excitatory or inhibitory, based upon our receptors. I say that because its release, potentiates stroke damage. Be careful. And sometimes, if there happens to be male impotence, it's treated by enhancing nitric oxide action. Thanks a lot, Sildenafil, also known as Viagra. Up next, I guess I'll touch carbon monoxide very briefly. That was brief. And then I'll now spend more time on the endocannabinoids. I did say that, the endocannabinoids. So these are what I call natural opiate neurotransmitters, and they make, of course, the endocannabinoids. So they act at the same receptors, such as tetrahydrocannabinoid, meaning, I'll say again, our brains make endocannabinoids that act at the same receptors as tetrahydrocannabinol. That's THC, you all, the active ingredient in marijuana. So what happens is they're common in that G protein pathway. And what they do is, like nitrous oxide, they're thought to be involved in learning and memory, but it's not wholly known. But in the very same way, they include, of course, a way to control appetite and suppress nausea. So I give you all of that to go to where I am nextly. The next thing I'll say is know those classifications of neurotransmitters, meaning how they are by class. And I'll say it this way. The effects can be excitatory or even inhibitory. Some that are excitatory, cause depolarization, period. If it's inhibitory, they cause hyperpolarization. And others, of course, have both those effects. So please keep in mind that. So, for an example, I'll give you. Amino acids GABA and glycine are usually inhibitory, whereas glutamate is typically excitatory. However, acetylcholine and norepinephrine each bind to at least two receptor types that cause, I repeat, opposite effects. Here's another example. Acetylcholine is excitatory at neuromuscular junctions. Ha ha ha, you all know this. However, acetylcholine is inhibitory in cardiac muscle. So for the future nurses listening, know your drugs, know the target, and of course know what receptors predominate, knowing their effects. So those actions can be, of course, direct or indirect. And I say this because a neurotransmitter that may act directly are those that bind to and open ion channels and give that rapid response, such as acetylcholine and amino acid neurotransmitters, typically direct acting. A neurotransmitter that is indirect may promote a broader, longer lasting effect, such as a secondary messenger, such as in that G protein pathway that you all learned about in your first biology course. So up next, there are disorders that are associated with neurotransmitter imbalances. I won't go through all of these at the moment, but things that you all should know about are right there before you. Clinical depression. It's real. And if you need help, please contact me. Call my office telephone number, and I'll pick up my cell phone and talk to you and find you a counselor that works here at Coastal Alabama Community College. I care about all of you all. Huntington's disease. Hypersomnia. The very same way. It could be insomnia. Even mania. Parkinson's disease, and tardive dyskinesia. These are all associated with an imbalance of a neurotransmitter. So these are drugs that alter neurotransmitter levels. I say you all should know a number of these. If you don't know them yet, begin learning some. And even in this class, we'll go through effective nicotine. On your test, I'll ask about Curare. I'll just be blunt. And what else is there? I guess that's enough there. But definitely nicotine and Curare. It's most amazing the effects of these and the mechanism of action they have. Please review this class. So what I'll now get to, meaning this class that you're seeing before you is what you know. So it says here, many neurons in the brain and spinal cord synthesize those neuropeptides. Just keep in mind that the enkephalines relieve pain sensations, and the beta endorphin relieves pain sensations, and substance P also does the very same. So I'm going to ask you about pain on your test. Please take time to learn. Up next it says opiates in the body. I won't spend much time here because of what I did earlier, but please take your time to go through this clinical application on opiates in the body and drug addiction. I did a bit of this moments ago. I'll do a little bit now to end the chapter. Clinical application 2.5. So drugs that alter neurotransmitters. on that postsynaptic neuron, either A, stop, or B, enhance synaptic transmission. So an antagonist is a drug that binds to the receptor and blocks neurotransmitter binding, as opposed to an agonist that activates the receptor, causing the action potential or aiding in the binding of a neurotransmitter. So repeated use of a drug causes a decrease in its receptors, so a person must take more of the drug to have the same effect. So that's, of course, what happens with amphetamines. They increase the norepinephrine activity, elevating that person's alertness and even mood. The person might stay up for days or even a week at a time. However, with nicotine, it binds to your nicotinic. I repeat, it binds to those nicotinic receptors. And I say those nicotinic receptors, they're nicotinic acetylcholine receptors. So these channels are for those positive ions to open, allow that influx, and of course leads to the feel-good neurotransmitter called dopamine to be released, leading to that pleasurable feeling. So with that, the person begins to, of course, want more and more and more and more and more. And it's called the hallmark of addiction. So as far as your test is concerned, please make sure you all can explain the events occurring to elicit a nerve impulse. That means you should know every piece and part that gets us to the overshoot. You should go through the voltage. You should know what the voltage is inside that cell throughout it all, from resting membrane potential to depolarization to threshold potential to, of course, repolarization to hyperpolarization back to resting membrane potential. And I say again, you should include every single piece of voltage and even those ions that are involved. They call them sodium and potassium. I should know the concentration inside the cell, the concentration outside the cell, the movement of those ions, the channels that are involved, and even the cause of this to even happen. I should also have in that answer what saltatory conduction is, how it occurs in the PNS by way of those Schwann cells and those nodes around VA. In other words, in your answer, it should be a full and complete answer, including sentences, not bullet points. And yes, you may draw, but of course a drawing shall not suffice to earn credit in and of itself. Words with the drawing. So if I can help you all out, please let me know. In the meantime, this is in the 10th chapter. This will conclude everything for Test 4. From here, we'll now get to, of course, the 12th chapter. Thank you all for listening. Please take notes well and study well. Hi, class. This is your instructor, Skylar Huff. So we're now here in the 12th chapter. So in chapter 12, this begins what is known as the special senses. So I chose to do the special senses here, and then, of course, we'll end the course with chapter 11. As it states, senses. As we are here in the senses we have, like I would say, control us. And when I say they control us, because, well, that's what they do. So as we are here, I'll take my time to ensure that I go through this and not go through this quickly at all, because it's important to know this, because you apply what you have here to, I would say, everything else in the course. So as we begin, sensory receptors, what they do is they take in what's in the world around us. So they respond to change in the environment, meaning those things called stimuli. And as I say it this way, we did senses before, those generalized senses, located, of course, by and large in the skin, even the joints, causing us to, of course, not fall down when we stand up. And even in our various organs, we have generalized receptors. Well, now we're getting special. So as I get to here, keep in mind that when I refer to the term sensation, that's someone becoming aware of that stimulus, meaning knowing it's there. And then perception is interpreting the meaning of that stimulus. So I guess I'll put it as being, let's go to ways to classify these sensory receptors. So they'll be classified by the type of stimulus they detect. They'll be classified by the classification by location, and then classified by receptor structure. So classifying in three ways: the detected stimulus, location of the body, and structural complexity. And of course, those that are special include things for sight, those for hearing, those for smell, and those for taste. Typical of the special senses are to be, of course, I say here, confined or restricted class to the area in the head. So, having done this, what I'll next get to is, of course, receptor types. You'll see this on your test. And I should stop now to mention something. This is final exam material. I repeat, this is final exam material. This is chapter 12, you all. So as we are here, I'll begin class with what are known as chemoreceptors. The chemoreceptors are quite special. Gotta love them. They respond to chemicals in some solution. I almost said an aqueous solution. Thanks a lot, chemistry. So the molecules that are smelled or even tasted. And keep in mind, class, even changes in blood or interstitial fluid chemistry. Chemoreceptors. Some of the most important things on earth are there. Let's go now to, of course, the pain receptors. They're also referred to as nociceptors, which is what you'll see on your test. So the nociceptors respond to potentially damaging stimuli. I repeat, things that are potentially damaging stimuli, such as that is called electrical, such as me getting shocked at the light switch on the wall. That is mechanical. Hard mechanical. And of course, thermal energy. Meaning I'll get to, I guess I'll say something, a story of which. I have hot water here at the house, and I'd imagine you all have hot water too. Gotta have hot water. But hot water, class, feels amazing, I say, up until a certain point. And you're saying, okay, so what do you mean? Well, when hot water is hot, well, I'll say it's hot. But as hot water begins to get hotter and hotter and hotter, it then becomes painful. So I'll put it this way. Heat from about 90 to 100 degrees Fahrenheit. And this leads me to what I call thermoreceptors, meaning your thermoreceptors respond to changes in temperatures. I'll say again from about 90 to 120 degrees Fahrenheit, and then again from about 50 to 140 degrees Fahrenheit. Thereafter, outside of this range, activates your nociceptors, and then it's painful. So when you carry that very, very cold, I guess you say, piece of meat from a freezer that's frozen, it begins to get painful. Or when I'm at the household, meaning my parents' household, and I turn that hot water on, I guarantee you it's well over 120 degrees Fahrenheit. Quite painful. Thanks a lot, nociceptors. I'll now get to, of course, what I call mechanoreceptors. The mechanoreceptors are very amazing too. I say this because they respond to mechanical force such as touch, pressure, and when I say pressure, this includes blood pressure. Thanks a lot, baroreceptors. Write that down, class. Baroreceptors, vibration, and stretch. Lastly are photoreceptors. They respond to light. And there are two types. Rods, cones, photoreceptors. So having done this, we've now, class, finished up the classification by stimulus type. I say, class, I cannot stress enough that you all should, I repeat, you all should review this and know this like the back of your hands. Because I just know that if you all can do this, you all will do quite well. Up next. It states here sensory impulses. Before I get there, I'll go back to location before I get there. So to classify our sensory receptors by location, they can be located, of course, externally. So they're called exteroceptors. That's E-X-T-E-R-O ceptors, C-E-P-T-O-R-S. So they respond to stimuli that arise outside the body. So most of these are at or near the body surface, including things for touch, pressure, pain, and temperature receptors in the skin. And even, of course, most of our special senses for vision, hearing, equilibrium, smell, and taste. Up next are interoceptors. And these are, I would say, so amazing because they're sometimes referred to as being visceral receptors because they respond to stimuli within the body. So it comes from our internal viscera and blood vessels. So with this, they include chemical changes, tissue stretch, and even temperature. So this, of course, is what causes us to feel A, pain, B, discomfort, C, hunger, or even, of course, D, thirst. Typical of which you don't even know what's happening there until, of course, something happens. And then lastly are proprioceptors. So they respond, to internal stimuli. So their location is restricted and they occur in skeletal muscles, tendons, joints, and ligaments. So this just monitors how organs contained, those receptors are stretched. So now that I've done this, we'll move on to the next part and piece. So I won't spend much time here. I'll get on down to, I've already done sensation and perception. I'll get to projection. So when projection occurs, that's just when the cerebral cortex interprets the sensation as it's being derived by those receptors, and then the brain projects the sensation to that apparent source and allowing you to know, A, that came from here, and that's what it was. Alright, so sensory adaptation. I'll make this quick. This happens quite quickly. And it's happening right now even, meaning you're wearing clothes. However, I doubt that you feel every article of clothing you wear. So this is important because we have to be able to ignore those unimportant stimuli, those continuous stimuli. Otherwise, you couldn't function. If you were feeling every article of clothing you're wearing, or hearing every little piece and every little part happening in the world around you. So it's just that decreased response to a particular stimulus from those receptors. So when this happens, I say it's quite amazing. And of course, it's best accomplished by thermoreceptors and our olfactory receptors. If you don't know what I mean, you may walk in and, of course, smell this scent or smell that scent. And then, of course, the scent disappears. Thanks a lot, olfactory receptors. And the same for thermoreceptors. The same for thermoreceptors. So I already mentioned, of course, these general senses with the exteroceptors, the interoceptors, and the proprioceptors. Please review these, class. And as I get to touch and pressure senses, this is all review. We did this back when we learned about skin and the skin and its derivatives, as you all, of course, gave the function of each of these parts. This is nothing new. I say you should review it, but nothing new at all. And of course, that's why they show you all the skin diagram yet again. I already spoke to this class by way of our thermoreceptors, meaning they are here detecting change. However, I said earlier, class, they're sensitive to things that are warm. So from about 77 to 120 or 113 Fahrenheit, above, it's nociceptors. The same for cold. At the approximate 50 degrees Fahrenheit, and of course to 68 degrees Fahrenheit, Anything class colder than 50 begins, of course, to trigger, to cause your nociceptors to activate. Hence, of course, it becomes pain, burning, and freezing. So let's go to pain. Pain is here. I say good. Why is it good? Because, of course, it alerts you to, of course, something that is potentially damaging to you. So with that, nervous tissue of the brain lacks pain receptors, although there could be referred pain there. And pain receptors are not ones to adapt, because as I said earlier, pain receptors, the sense of pain, it keeps us alive. So as I mentioned perception of pain, what happens is histamine is released, potassium is released, ATP is released, even acids and bradykinins, meaning those are some of the most, or among the most potent pain-producing chemicals. So, of course, if you've ever cut your finger, You may have noticed that you first feel that sharp, sharp pain. And of course, by later burning and even aching. That sharp pain is carried by those smallest myelinated sensory fibers. The fibers class can carry nerve impulses at speeds upwards of 300 miles an hour. If that's not fast, I don't know what fast is. So, of course, thereafter, glutamate and substance P is released. And, of course, that should allow you to do what I call be able to take some things. So, if you cut your finger while fighting off an attacker, you might not notice that cut at all. You say, why? How? It's all because of what I just taught you all class earlier. It's all because of your body's endogenous opioids, meaning those pain-suppressing analgesic systems, the endorphins, the enkephalins. They play that key role, as I told that story earlier. So, having known this, I hope you all know a lot more about pain. Before I get to referred pain, I want to mention pain tolerance. So keep this in mind and look for it in your test. All of us, we all class, have the same pain threshold. Or in other words, we all begin to perceive pain at roughly that same similar intensity. However, what is different, what does vary, is our tolerance to pain. Hence, of course, somebody may say, ooh, I'm sensitive to pain. Ooh, that hurt. And you might say, that didn't hurt. It just may be that you can tolerate, of course, more pain than him or her. Up next, it refers to visceral pain, and this comes from the visceral organs themselves. Please look at the diagram about referred pain, you all. As I mentioned referred pain, this is just meaning that pain, that visceral pain, may feel as it's coming from some other body part than, of course, the place it actually has come from. Hence, of course, it's called referred pain. One that may save a life is that heart pain often feels like it's coming from that left shoulder or even the middle portion of the arm and even down to the pinky finger. And it's all heart pain. Hence, it's referred to as being referred pain. It's all because that visceral pain afferents along the same pathways as somatic pain fibers, which of course is why it's being referred to another body part. And having done this, I guess what I'll get to nextly, and there's a referred pain diagram, and it does show you what I mentioned about pain in the heart being referred, even down to, I say again, that pinky finger. So please take note of that. It could save a life. It could most definitely save a life. And this is showing how that pain is referred. I believe what I'll do nextly is I'll say please review how pain is treated. NSAIDs are good for pain, but they can cause problems, class, with your gastrointestinal tract. That's why you've likely been told to take some NSAID, that non-steroidal anti-inflammatory drug, and then take acetaminophen. I say again, you may have been told to take the NSAID, the non-steroidal anti-inflammatory drug, and then take acetaminophen. To, of course, alternate, because yes, the NSAID, such as aspirin or ibuprofen, can really cause issues with your gastrointestinal tract. But on the converse, acetaminophen, although it does not affect the GI tract, it can damage your liver. So please be safe in taking these pain medications. Please do not just take Tylenol, which of course is the brand name for acetaminophen. And please do not just take an aspirin or an ibuprofen, because both of those can cause damage to the body. I'll read about phantom limb pain, because it is real, and it deals with, of course, an amputation. So if you have a limb being amputated, what could very well be done is to use general anesthesia or an epidural block to block neurotransmission just before, of course, the limb has been amputated, to ensure that doesn't occur. Special census. So I believe this will be a good place to stop and lecture for today. And we'll begin the next time we meet with the special census. So please take notes, class. This is the beginning of the course, the final exam. And if you need help, feel free to call or email me. Have a great day, and thank you all for listening.